Edu Arctic2, Polar Research for Education, innovative program in Poland and Norway. Webinars.
Norway, it's very dark outside here because we don't have the sun anymore. The sun doesn't come up over the horizon. So I'm very excited that so many of you want to listen to this presentation. And today I will talk a bit about genetics and how we can use greetings from rainy Greece. Hi. Welcome. And today I talk a bit about genetics and how we are using this up here to study freshwater organisms, mainly fish species. But before we do this, I thought I'd introduce myself to those of you who don't know me very well yet. So I'm a scientist and I've studied biology, and I work at Svanhof. That's a biological station which is situated up here in the north, very close to the border to Russia and to Finland. And the station is about 400 kilometers north of the Arctic Circle. And just to give you a few impressions here, on the left-hand side, it's a picture of the station with the botanical garden in the front, and up here you have a picture where you see the station, the river, the Pacific River, which we'll talk today about, and you can see Russia here in the background. So it's really, really close. And we're working on quite a number of different species, as you can see here at the bottom. And fish species are one of those, and freshwater systems like rivers and lakes. And the reason for this is that they are very important for the local communities, for fishing, for angling, for tourism, and for us, for research, to understand a bit better what's going on in rivers and lakes. And I just thought I will give you sort of a quick run through what we will be talking today about. And before I start, I think Hilde wrote this already in the chat. You can ask questions in the chat anytime. So please feel free to do this, especially if there are classes attending, that if you have questions, something is not clear, or you have other questions, please feel free to ask questions in the chat. So I will introduce you a bit to what actually genetics is, what is this all about, and we'll then go through a few terms I think are probably important that you get them right in your head. So that's, I think, very important. But then the second part of the presentation, I will talk a bit more what we are doing with genetics and DNA analysis. So I will give you a real-life example and a few concepts that we are dealing with and we are interested in understanding better in fish species. And at the end, also to let you know here, I have prepared a Kahoot, and for those of you that still have time, please take out your phones, mobile devices, and PC or whatever you have at hand. And then we will have a little Kahoot about the presentation, so as a nice little quiz at the end. And then I just want to prepare you that I have prepared a question also at sort of the end, towards the end of the presentation, about the research we are doing. And I would be interested in knowing if you think this is working or not. You will see more detail later on. But just to prepare yourself that you can either unmute and give comments, or that you write into the chat again if you have comments regarding this. Okay, so let's get started. And I think the easiest way to understand what actually genetics deals with is if you're in a classroom setting right now, that you look at each other. You're all very unique. You all look different, obviously. You have different, except if you're twins, but you'll all look a bit different. You have different hair colors and eye colors and other characteristics. And those characteristics in your appearance is the phenotype. Phenotype means simply what we can see on the outside. And So phenotypes is what we are looking at in genetics, so what we are seeing. So in animals, obviously, we can have they can have different size or the colors as well. So we are often interested in understanding why there are differences. How can we explain this? However, obviously, all of us have a mom and a dad, so our parents have given us these characteristics, these traits, the different hair color, the different eye color, and so on. So there's inheritance involved in genetics. And then in genetics, we are interested to understand what are the molecules, who is carrying this information, what is the unit of inheritance, how is this transferred from one generation to the next. And the carrier of information are, of course, genes and the genome. And the genome is located in each of your cells in the cell core on the cell nucleus, here shown in this purple pinkish color. And this is really where all the information is stored. There's one exception to this. You have other organelles in the cell called mitochondria, and they are important for energy production, and they have their own genome. And but most of the genome and genomic information is stored in the cell nucleus. So this is where we find then chromosomes and the DNA. To discuss and introduce you a bit more to this, there are a few more terms you need to know, and I think the best comparison is actually to think about the genome and DNA and these different structures if you compare this to a book. Because the core molecule in genetics is, of course, DNA, and DNA stands for deoxyribonucleic acid. Complicated word, but it's basically a structure that is made up of 4 molecules, which are called adenine, cytosine, guanine, and thymine. And those 4 nucleotides, those 4 molecules, are basically the alphabet of your genome. They code in the succession and the combinations they can make up. They are making up different words. So basically, if you compare this to a book, you need an alphabet to write a book and to write words. And in DNA, your alphabet consists of A, G, T, and C. And these molecules are organized in a double strand. There are 2 strands here, 1 and 2, which we call the double helix, DNA double helix. And the next thing to think about, okay, then we have sentences. Words, several words make up a sentence. For example, genetics at school, of course. And the same applies to the genome. So the succession of this alphabet makes sense. It codes for a specific information, usually proteins. So everything is basically translated into protein in the process. And what's also important to realize here that sometimes mistakes happen in a cell, and this can lead to nonsensical information on the genetic side. So there may be mistakes. But also, again, it's very similar to if you make a typo in a sentence. So genetic is coal or genetic is wool doesn't really make sense. So it's, again, very, very similar to what you know if you're thinking about books and what can happen there. And the next bigger unit you have are probably chapters in a book. These are larger sections, they are larger collections of sentences. And chromosomes are structures. Basically, these are larger collections of genes. So basically, it's a compact form to pack all the genetic information into a cell. And this is needed because otherwise it would really literally not fit. It's a very, very dense form of packaging DNA. And finally, we have the genome. In the book sense, it's the whole book. It contains all the information that it's needed for the story. And the genome is an all the collection of all the genes, all the chromosomes in the cell that contains all the information for all the functions in the body. And by the way, here on the right-hand side, you see all the chromosomes that humans have, which we call also a karyotype. So karyotype is basically lining up all these chromosomes that we find in the human cell or in other cells as well from other animals. In the human case in particular, you have 23 chromosome pairs. So, and one of those chromosomes pairs are called sex chromosomes because it's the X and Y in this case. So it's a male that determine the sex of an organism. And so if you're female or male, a woman or a man. And the other 22 pairs are called autosomes, and they have all the other information basically. Okay, so this gives you sort of a tiny introduction, a very crash course into genetics, but basically these are sort of some of the fundamental units and terms that are important to know. And finally, I wanted to introduce you to what a mutation is. And this is basically a typo. I talked about this a bit earlier, the typo in the sentence, that it doesn't make sense. And a mutation is basically a change of this DNA sequence. And here I have 3 DNA sequences. And you see I have highlighted them that a change occurred. And this change is called then a mutation. And sometimes, this variation is basically the cause for different morphologies or phenotypes again. So in this case, I took 3 imaginary fish that have 3 different colors, and the combination of these changes causes the change in the phenotype. And this is really the essence what we are interested in. We're really trying to understand what mutations, what changes are connected to different behaviors or different morphological characteristics. We are very interested in this. And of course, some of these you can also say that these mutations, for example, are important in disease genetics and medicine to understand why disease develops. So this is a very active, ongoing research field as well. So just to give you a few ideas why we are so interested in understanding what these mutations, what these changes mean for us. And this is really, really the absolute basic interest we have. And because genetics, because of technical advances in recent, probably the last 2 decades, and with the recent event of genomics, so we are really not studying parts of the genome now, we are studying whole genomes of organisms. And this is because of technical advances in recent years, probably last 7 years now for genomics, 7, 8 years. We apply genetics and genomics to a multitude, to numerous fields. So just to give you a few examples, I'm sure that you have seen a lot of CSI and criminal TV series. Of course, forensics is a big field. And we're using this also in wildlife forensics. So for example, if you're interested, if someone illegally hunted reindeer up in the north. In northern Norway, it's very interesting. We have the so-called reindeer police, and they take cases on like this, and they contact, for example, us to do the genetic analysis to tell them if someone hunted a specific reindeer. And there are a lot of other examples, of course. Medicine, I already mentioned, a lot of disease genetics, cancer genetics, and of course, very, very recently, we're all sitting in Zoom meetings and on Skype and have webinars instead of meeting in person. It's, of course, COVID. The COVID genome has been sequenced very, very rapidly, and it's one of the reasons why we know more about this disease. And then a bit closer to home, what we are doing up here at the station. We are looking into biodiversity, so we're interested in different species and how we can explain this diversity. Then functional genetics, this is really to understand what a gene does. And surprisingly, although you could think that we know a lot about the human genomes, for example, we do not know the function, what a gene does in a lot of cases. So there's still a lot of research to do. And then we have population conservation genetics. And I have a further slide. The next slide, I will talk a bit more about this. But basically, we are trying to monitor animals and understand how many of them are they, and if they if they have offspring, so if they have young individuals in the population, and which sex they have, so that we understand, for example, how many males and females are in the population. And in some cases, and I will talk about this a bit more in the next slides, we have cases where we help the natural population, the populations in the wild, by breeding individuals and then releasing them. But there are certain problems associated, linked to this, and I will talk a bit about that. And in connection to this, sometimes we are interested to understand sort of the family structures. This can be in humans, it can be interesting in medicine. If you try to understand how a disease, for example, is inherited, or what the risk is that your kids could inherit a disease, for example. In wildlife genetics, it is more that we are interested in understanding who is probably producing more offspring. It could be interesting in some species to know where this suspicion is that only a few individuals actually produce offspring for the next generation. So these are not all fields, so they are far more, and it's ever expanding. Genetics is a great subject to get into, and the applications are ever expanding. And I told you already that what we are doing up here is often connected to a field called conservation genetics. And conservation genetics is really to understand how we can protect endangered species. So obviously the panda is a species that is endangered. It could go extinct if we are not really closely having an eye on the remaining individuals. And the same, for example, in Europe, the wolf populations and other large carnivore populations like the brown bear need close monitoring and management to really make sure that we keep those populations and species alive. And the basic analysis we are doing for a lot of our projects up here is that we take a sample. Often we use actually samples like feathers or poop, actually, scat samples, or hair. We can also use scales from fish, or sometimes a tissue sample. And then we extract DNA in the laboratory, and we have a sort of a magic machine that basically determines the sequence of those nucleotides, of the T, A, G, T, C, and so on. And then with computer programs, we can basically have a look and align them and then see if there are certain mutations occurring in certain places. And this can be used then to study wildlife populations. So this is the basic principle of what we are doing. But I thought it's also interesting to give you a real life example. So we have talked a lot about what genetic is and what you can do with it. And I thought I'd talk a bit about brown trout populations. It's a fish species which we have in the Pacific River system I mentioned earlier up here. And that river system is quite interesting because over the last probably 60 years or so, 60, 70 years, 7 dams were built into this river system. And at the time, people thought it's a great idea to for energy production to use hydroelectric power. However, people didn't think so much about what would it do to the populations, the fish populations that live in these rivers. And one of the effects that can be is that some of these migrating fish species, like salmon species and also brown trout, they can't migrate to their spawning, to their reproduction areas in the river system. And over time, if this happens for a long time, then what can happen is that they're really becoming isolated. And this is not very good because some of these populations might suffer. The other problem is that in a lot of rivers, either the local communities go for fishing, for food, literally, but we have also tourists coming up that do recreational fishing. So different pressures, human activities put a lot of pressure on this system, on this river system, and therefore it could be that these fish populations and also other populations that are connected to the river suffer, and that they probably have problems to survive in the long term. So this is sort of a bit of the background there. And because of these potentially separating effect of these dams, there has been a breeding program initiated. And the idea is that brown trout is bred in a facility, and about 5,000 fish are released every year in different sections. So the idea is that the effect, the isolating effect of the dam is probably diminished by this. And what you need also to know is that on the Russian side, there are 3 sections, which are A, B, and C here. There's no stocking. This is because the river basically constitutes the border between Norway and Russia, but there's one specific part that's only in Russia. And Russia, as a country, has decided not to stock, whereas Norway releases those fish specimens in this part from D to J. And what is interesting for us is to see how does this affect actually these populations, because basically it's a natural experiment we have, that we have a setting where we have 2 groups that do different things. In this case, 2 different countries have different management strategies for this river system. So basically in our research, we were interested in 2 questions. Do these dams separate fish populations in different river sections? And since some parts are in some parts fish are released every year, does this, what we call stocking, change the genetic composition, the genetic makeup of those fish? What happens there? And here, just on the left-hand side, it's a brown trout, so that you have also seen the species. And what we find there is that indeed there is a difference. So here on the left-hand side, it looks like a complicated figure, but the only thing you really have to see here is that these river sections, B and C in particular, that are in Russia, have a different color, which means in this case a different genetic composition, a different genetic makeup, to largely those other sections. What this tells us is that there is genetic differentiation. These stocked fish on the Norwegian or on this D to J section where fish are released have a different genetic makeup than the others. And there's an effect known that if you breed fish over generations, that you can actually change the genetic composition. So something has happened here. And the other interesting result of this research was that in the Russian part, remember that I said there's no stocking, so no fish are released. There we find between two of these sections a subtle genetic differentiation. Again, the two different colors basically indicate you have more orange here in the C section. This indicates that they are genetically differentiated. And this is most likely because you have a dam there and no individuals can migrate back and forth. And what we are seeing, we're starting to seeing a differentiation in the genetics of those two fish that are really, really close together, adjacent, neighboring sections, but they are separated. So there's also an effect of dam structures in parts of the rivers that do not receive stocked fish. And one of the effects that we did see, what was sort of surprising in a way, is that in this blue section here, they are all very similar genetically. And what the breeding most likely introduced is what we call a bottleneck. And the bottleneck basically means that you select specific individuals from the whole population and then you use them in the breeding program. And what this led to is that we are losing some of the variation. In this case, these reddish alleles. So if these are all your different genetic variation traits, green, this pinkish color, and this red, you see that we are losing this red ball, which simply indicates genetic variation here. So we are losing something in this bottleneck. And bottleneck events also occur in nature. For example, if some individuals are going to different areas, so only a few individuals with specific genetic variation are forming a new population, for example, somewhere, then you can have this as well. So basically what we see is also that in this case, you only have two green genetic variation balls here. So it can also that the proportion or frequency of specific genetic traits will increase or decrease in this surviving population. And what we think has happened here with the breeding, that something like this has happened. So we see that from the genetics that they are undergoing a bottleneck, that these different proportions of genetic variation is changing. So definitely there's something going on, and we need to, of course, think about if this is the best way of dealing with the problems we have in the river, and how do we want to protect those fish species. And just on a side note, in the next study, we looked at the tributaries, sort of the side arms of the main river. And there, again, it looks like a complicated figure, but basically here it's every different color means there different genetic variation. What you see, it looks far more colorful. So we have far more genetic variation in those side arms of the river, or tributaries. And what we see also is that those side arms are genetically different from the main river to a large degree at least, and also from each other. So there seems to be a lot of genetic variation hidden in those tributaries. And one of the questions we are interested in is would the main river look actually more like this if we wouldn't have the stocking? So are we making it better or worse actually with the release of those bred fish specimens? And I promised you that I have a question for you. So I would like to know, with what I have presented here about the breeding program and the genetics, do you think this breeding program is a success? Do you think it works? Or do you think something else should be done? And if so, what do you think this should be? So I would be interested, and if you want to write in the chat, what do you think? What's your feeling about the status of the river and the brown trout population? So you can write into the chat if you want. I will give it a few minutes, then we have to move on probably to make sure that we can also do the Kahoot and so on. But let me know if you have any thoughts on that. Okay, a question from Lucas is, what is the breeding program impact on jobs and the economy? That's a very, very good question, because we are working here at the biological station with the local communities, and obviously it's there are sometimes, yeah, let's say different interests from different groups. And the breeding program is provided by one of the power station companies. So they actually putting some money into the breeding program. And so certainly there are some jobs connected. However, it is also important to see that the breeding program, because you have a lot of tourism and a lot of Norwegians, but also international guests come up here for recreational fishing. So you have a lot of secondary impacts on hotels, for example, or other service industries like restaurants and so on. Boat lenders, whatever, fish licensing, also an income source. So the river is very, very important for the local communities. And connected to this, of course, also the breeding program is very, very important to keep those jobs in these different industries. So it is definitely an important source of income for the local communities. I don't have exact numbers, but certainly it's a very important part for the local communities. Any other questions or ideas? No one dares to speak up? What do you think? No, I think you have to say, the question is, do you think the impact on the jobs and economy determines if it's a success? It's part of this, but I'm a biologist, I'm concerned about the well-being of the fish populations and how healthy the river system is. Because if some of these fish species disappear, for example, the problem is then that the river system health is probably also affected. So some species are dependent on the brown trout in the ecosystem to function, for example. So are we doing a good job here if we manage to lower the effects of the dams? but we change the genetic composition of the fish species. Is this a long-term viable strategy? It's an interesting thing. It's not an easy thing to answer, actually, right? So because we have these different interests and different groups, there's a question by Kipa. Did it cause overpopulation in some areas regarding the fish? Definitely not, because the annual breeding program with the release of 5,000 fish is basically necessary to keep enough fish for the recreational anglers because of this tourism. We have a lot of anglers come up here. So it's really important to keep for the population, for the fish population, to have enough numbers. Okay, so probably you can, the idea of this question was basically to make you think a bit about what this actually means. So we're doing this research, but why and how this is connected. So there were actually quite a few good questions here. Also probably things I could have discussed in more detail. However, I will plug in here that we have another presentation about invasive fish species, and I talk about the economic impacts, for example, for local industries there as well. So you can check this, as we are doing this 2 more times, the other talk. There's a question from Alexandra. I think that damage made by disappearing population would be greater than changing the genotype. And this is, of course, true. The problem with that is, though, I told you that each gene has, and each mutation can change the function of a gene. So what we are seeing, for example, in those bred fish is a domestication effect. What this means is that those fish that are in this breeding facility, that they show characteristics in the phenotype and in the behavior that make them less likely to survive in the wild, for example. So and a lot of studies have been done because some of these fish are just not used to find food because they are fed in these facilities. So they know that they are fed, so they don't survive very well. And the problem with that is that then they don't reproduce, and then, or not as much as you would hope for, and then you have to continue this breeding program. There are also other effects. Sometimes there are behavioral changes. So some studies point to that these bred fish are very, very aggressive, and it's most likely a density problem because they're in these water tanks and these basins. There are a lot of fish crammed into those water tanks. What happens is that they develop a very aggressive behavior. What happens then in the wild is that they are also very aggressive towards the wild fish. And this can lead to changes how actually the biology functions in the river systems. So there are other problems, and though a lot of these changes go back also to genetic changes. So it's sort of a mix of environmental conditions, but also genetics. So the breeding program can change some of the gene functions, and with this, some of the behaviors, for example. So, but in general, I agree. That's, of course, the aim of the breeding program is to keep the species alive until we have either better tools or understand the system better. But of course, once we know more, there's always room for improvement, for example, for the breeding program. Okay, so I think we will move on, but thank you so much for all the questions. This was great. So it's always good to have a bit of interaction. Here I basically then summarized very, very coarsely. We have actually discussed more, but just very close to the examples I have given, that the breeding program can actually prevent the isolating effects of the dams. However, we are losing genetic variation in this process, and this can be a problem in the long term for several reasons. Here, we are discussing actually with the breeding facility how we can improve the breeding practices. So, for example, can we take more fish in so that we have a better representation of the genetic diversity of those fish in the breeding facility every year? So this was one recommendation, a very practical management conclusion or recommendation we made. And of course, other factors could play a role. Can we increase natural reproduction in the river? So should we think about restoration of certain areas? So these fish need certain spawning areas, reproduction areas in the rivers. So they have specific requirements for reproduction. Can we work with this? Can we improve that part to make this sort of conservation and management program better? So these are just a few examples how genetic research then connects actually to real life example and to real management questions, and how we can use this knowledge to improve things that are done in breeding programs, for example. We're doing this with other species up here, but I thought in this case we talk about fish, so it's a good example. Okay, so don't hesitate to ask more questions in the chat if it comes up. However, if you don't have any questions right now, then I would suggest that we go to the Kahoot. But I give you probably a minute if you have any other questions regarding the entire presentation here.
Watch other recordings from webinars on our YouTube channel, youtube.com/EduArctic. Edu Arctic2, from polar research to scientific passion, innovative nature education in Poland and Norway receives a grant of 240,000 euros received from Iceland, Liechtenstein and Norway under EEA funds.